Good morning. Our lecture today is early tooth development. Dentition. In human, we have two sets of dentition: primary dentition, or deciduous teeth, developed during a prenatal period in the intrauterine life. It includes 20 teeth, 10 in the upper jaw and 10 in the lower. And second dentition is the permanent dentition, develops as the jaw grows and mature. This includes 32 teeth, 16 in the upper and 16 in the lower jaw. And we have mixed period or mixed dentition where we have deciduous and permanent teeth in the same time. This period. Tooth development is the complex process by which teeth from the embryonic cells grow and erupt into the mouth. So the development of the teeth starts during the embryonic development of the embryo and continues till the eruption of teeth inside the oral cavity. This includes deciduous and permanent teeth. Tooth morphogenesis is an advanced process that is regulated by sequential and reciprocal interaction between the epithelium, which is cover the surface of the oral cavity, and the layers below the epithelium, which is called mesenchymal tissue or connective tissue. The dental organ or tooth germ, which that form the tooth in the future, is the term used to constitute the structures that has enamel organ, dental papilla, and dental follicle or dental sac. So the meaning of tooth germ is the primitive of the tooth, which include the three structure, which is the enamel organ, which give rise in the future the enamel structure of the tooth, dental papilla, which give rise to the dentine and the pulp of the tooth, and dental follicle or dental sac, which formed the periodontal ligament, cementum, and alveolar process. Tooth development. In this picture, we have in A, this is the bud stage. In B, this is the cup stage of tooth development. C is the bell stage. And in D, we have the dentinogenesis, meaning the dentine formation, and aminogenesis, which is the enamel formation. In E, the crown formation. And in F, we have also a crown formation, completion. In G, there's a root formation and eruption of the tooth inside the oral cavity. In H, the function of this tooth in the oral cavity by mastication. So this period A, B, C, D, and E, and F, this inside the jaw. This is the oral epithelium, and this is the connective tissue. We have this teeth during growing from the embryogenesis till the tooth inside the oral cavity in eruption here, and this is during function. So it's the complex process. The primitive oral cavity, or called stomatodeum or stomodium, is lined by stratified squamous epithelium called the oral ectoderm or primitive oral epithelium. So the oral cavity or primitive oral cavity during embryogenesis is composed of the surface layer, which is originated from the ectoderm called oral epithelium, and the layer below the oral epithelium, which is from the mesoderm. This mesoderm have the neural crystals, which come from the periphery of the neural tube during development of the neural tube. These cells the neural crystals migrate to the mesoderm below the ectoderm, and the structure which is present here is called ectomesenchymal cells. These ectomesenchymal cells will form the dental papilla and dental sac, while the oral epithelium, which originates from the ectoderm, will form the enamel organ. Three structures form the tooth gel. The oral ectoderm contact the endoderm of the foregut. The foregut is lined by endoderm, while the oral cavity is lined by oral ectoderm. These two-layer ectoderm and endoderm fuse together and form buccopharyngeal membrane, which separate the oral cavity than the foregut. At about the 27 days of gestation or embryogenesis, this membrane, buccopharyngeal membrane, rupture, and the primitive oral cavity establish a connection with the foregut. So this membrane is ruptured at about 27 days or 1 month of intrauterine life. Most of the connective tissue cells underlying the oral ectoderm, as we said, are of a neural crest or ectomesenchyme in origin. Primary epithelial bond. 2 to 3 weeks after the rupture of the buccopharyngeal membrane, as we said before, when the embryo is about 6 weeks of intrauterine life, certain area of basal cell layer of oral epithelium or oral ectoderm proliferate more rapidly than do the cells of the adjacent area. This leads to the formation of the structure called primary epithelial band. This is the primary epithelial band in the upper jaw, and this is the primary epithelial band in the lower jaw. This represents the thickening in this layer of oral epithelium, and this thickening is proliferation of basal cell layer of oral epithelium that cover the oral cavity. In certain area, this formation the primary epithelial band, which is the band of epithelium that has invaded the underlying tissue or ectomesenchymal tissue along each of the horseshoe shape, like the horseshoe shape in the future dental arch. This represents the future maxilla, and this represents the future mandible. This is thickening like the horseshoe. Each of this primary epithelial band will give subdivision. Lingually is the dental lamina, and labially is the vestibular lamina. So this diagram represents the development of a primary epithelial band and dental lamina and vestibular lamina. At 6 weeks of intrauterine life, beginning of the tooth formation or the formation of the tooth germ. There's the formation of primary epithelial band, as we said, about 6 weeks. Then at 7 weeks of primary epithelial band will give two subdivision, dental lamina and vestibular lamina. Vestibular lamina will give later oral vestibule, which separate, or the sulcus, separate the lip and cheek from the teeth-bearing area, while the dental lamina is the very important structure which that form the enamel organ, and then the formation of enamel and interaction with the under structure, the ectomesenchymal structure, and form a dental papilla and dental sac. All deciduous teeth and all permanent teeth will form from this dental lamina. So this is the very important structure. Lingual extension of the deciduous teeth will form the permanent teeth. Every deciduous teeth have lingual extension which formed the permanent successor. central incisor deciduous will have a lingual extension of permanent central incisor and so on till the permanent premolar, while the distal extension of dental lamina will form the permanent molars, first, second, and third permanent molar. Tooth formation initial stage, as we said, we have this is the oral epithelium, and this is the proliferation of this oral epithelium toward the ectomesenchymal cells here and form the dental lamina, which is this proliferation in this side. This the lingual extension of the primary epithelial band here form the dental lamina, and the labial extension form vestibular lamina, which later form the vestibule. While this dental lamina from this side will form the later on will form the enamel organ, while this ectomesenchymal cells will form the dental papilla and dental sac. These three structures will form the tooth germ. Dental lamina. Dental lamina appear as a thickening of the oral epithelium adjacent to the condensation of the ectomesenchyme. This is the oral epithelium, which line all the oral cavity. This is the tongue, also lined by oral epithelium, and this is the mesoderm. And this is the oral epithelium, and this is the mesoderm. Here we have thickening of the primary epithelial band, which later give on the dental lamina, and this is the dental lamina is proliferate toward the ectomesenchymal cells. Between the oral epithelium and ectomesenchymal cells, we have the basement membrane here. 20 area of enlargement or knob appear, which will form tooth buds for the 20 primary teeth. So in this thickening and the formation of dental lamina will give rise the deciduous teeth, and later on from lingual extension of dental lamina will give rise to the permanent teeth. So we have a 20 area of thickening with the formation of 10 dental lamina, and from this dental lamina we have the formation of 10 in the upper and 10 in the lower jaw as the deciduous teeth, and then a permanent successor. At this early stage, the tooth buds have already determined their crown morphology. Succession lamina or dental lamina of permanent successor will form which permanent teeth developed. So we have in the tooth development, we have two stages, morphological stages and physiological process or stages. Morphological stages depend on the morphology of the enamel organ of the tooth germ. So the morphological stages of tooth development include bud stage, when the enamel organ is like the bud; cup stage, when the enamel organ like the cup; and bell stage, when the enamel organ represents as bell in shape. Bell stage, we have two subdivisions, early bell stage and advanced or late bell stage. Early and advanced is different by the formation of the hard tissue like enamel and dentine. So in early bell stage, we didn't have dentine and enamel formation yet, while in advanced bell stage, we have the formation, beginning of the formation of dentine and the enamel, and later on when the crown is complete, the formation of the cementum of the root and dentine of the root, and formation of periodontal ligament and alveolar process. While the physiological process include the initiation, the beginning of the formation of dental lamina, a proliferation which is occur in bud stage and cup stage by the formation of a new cell layer and mitotic division. Histodifferentiation is occur in the bell stage when the cells is mature and formed a new cell layer, when the inner enamel epithelium will form a mesoblast, and cells of dental papilla form the odontoblast that form the dentine. Morphodifferentiation means the morphology of the tooth. We have anterior tooth with one cusp and posterior tooth with 2 or 3 cusps, and so on. The fifth stage of physiological process is the opposition, is the formation of the hard tissue like the dentine, enamel, cementum, and bone. So this diagram shows the morphological stage and physiological stage interaction. Dental lamina is the initiation. Body stage, cup stage is the proliferation. Early bell stage, histodifferentiation is the formation of odontoblast, ameloblast, and so on. Advanced bell stage is morphodifferentiation, the crown formation, complete crown formation, anterior or posterior teeth. And 6 is the formation of enamel and dentine matrix, which means the opposition physiological stage. This diagram shows the head of the embryo during early tooth development. This is the eye of the embryo, and this is the represent the future nasal septum, and this is represent the primitive oral cavity. When we take this cube, this represents the oral epithelium at this area, white pink, and this represents the mesoderm or ectomesenchymal cells. This is the beginning of the thickening of the primary epithelial band, which later give rise to the dental lamina, proliferation and thickening here, and this proliferation continue toward the ectomesenchyme and the formation of the enamel organ like bud. So this represent the bud stage of tooth development, and this is the ectomesenchymal condensation of these cells to form the dental papilla and dental sac. This proliferation continue till the formation of the morphological stage of the enamel organ like the cup. This is the dental lamina, and this is the oral epithelium, and this blue represent the dental papilla, which cover by dental follicle or dental sac. So enamel organ, dental papilla, dental sac or follicle. This represent a tooth germ. This is the bell stage when the enamel organ like the bell, and this is the dental papilla and covered by dental sac with the enamel organ, and this represent the dental lamina and oral epithelium. Erupted tooth when the crown is complete and the root is formed, the tooth is erupted inside the oral cavity and then function. This diagram represents the two layer, as we said before, the oral epithelium, green one, and the brown is the mesoderm, and the dark brown represent the ectomesenchymal cells. The oral epithelium begin to thickening in 10 area in the upper and 10 area in the lower. This represents the dental lamina at about 7 weeks of intrauterine life, and this is the thickening in the ectomesenchyme, which ectomesenchyme have the neural crystals inside the mesoderm, and this later on represent the dental papilla, and both of them, dental papilla and enamel organ, is covered by the dental follicle or dental sac. This oral epithelium, dental lamina, and this is the enamel organ here. This is the histological section represent the oral epithelium, which cover the oral cavity. This is the space of the oral cavity, white color. This is the tongue, also covered by oral epithelium. And this is the connective tissue below the oral epithelium, which come from the mesoderm. And this is the ectomesenchymal cells, because the neurolicral cells migrate here and form the ectomesenchyme. This is represent the future bone of the mandible. This is represent the Meckel's cartilage, present only in the mandible and surrounded by bone. This is bone trabeculae future, which form the future mandible, and this is the tongue. Here we have the dental lamina proliferation toward the ectomesenchyme, and the end of the dental lamina represent the enamel organ, which look like a bud in shape. This is the enamel organ, and this is the condensation of the ectomesenchymal cells here to form the dental papilla and dental sac. This high magnification of a previous slide, also histological slide, this represents the oral epithelium. This is the basal cell layer, thickening and form the dental lamina here. The end of the dental lamina is the enamel organ. Enamel organ in body stage histologically is formed from the cuboidal outer or peripheral cells. Cuboidal single cell layer surround the enamel organ in body stage, and the central cells is the polygonal cells present inside this enamel organ in body stage, and this is the ectomesenchymal condensation to form dental papilla. Between dental papilla and the peripheral cells of the enamel organ, there is a junction, is the basement membrane. Later on, the proliferation is continued of the enamel organ. So there is invagination in this area from the pressure from the dental papilla toward the enamel organ, and also here the proliferation of the enamel organ toward the dental papilla. So the enamel organ morphologically represent the cup. So the enamel organ look like the cup, so the stage is called cup stage. This is the oral epithelium present, and this is the dental lamina fuse the oral epithelium with the enamel organ. This is the enamel organ in cup stage, and this is the dental papilla here adjacent to the enamel organ, and the outer layer is the dental follicle or dental sac. Both dental sac and dental papilla from the ectomesenchymal cells, while the origin of the enamel organ is from the oral epithelium. Why from the oral epithelium? Because the dental lamina is from the oral epithelium, and the oral epithelium is from the ectoderm. This is another histological section of the cup stage. Why we call it cup stage? Because the enamel organ look like cup. This is the enamel organ fused with the dental lamina, which attached to the oral epithelium. This is space is the oral cavity, and this is the tongue of the embryo, and this is the lip, lower lip, and this is the vestibule or sulcus that separate this lip from the future teeth present here. And this is the, as we said before, Meckel's cartilage only present in the lower jaw, surrounded by developing bone of the mandible. Higher magnification of a previous slide. In the cup stage of tooth development, this is the enamel organ in cup stage, and this is the dental lamina, and this is oral epithelium. This is the space of the oral cavity. In the enamel organ, histologically in cup stage is formed from a 3-cell layer. The outer cell layer is present here, present in the convexity of the enamel organ, is called outer enamel epithelium, which composed of cuboidal single cells attached to each other by desmosome. This is the outer enamel epithelium. And in the invagination area, the cell layer is called inner enamel epithelium, which is columnar in shape, short columnar in shape, also single cell layer attached to each other by desmosome. This later on differentiate to ameloblast that give rise to the enamel structure. And the third layer of the enamel organ in cup stage is the stellate reticulum. Stellate reticulum cells of this area is a star in shape, have the intercellular process attached to each other and form intercellular bridges. Between these bridges there's a space which have glycogen, and this cells is the thicker cells present in the enamel organ. Here we have the dental papilla, which is composed of condensation of cells of ectomesenchymal in origin, and surrounded by the enamel organ and dental papilla by also cell from the ectomesenchyme, which is called dental sac. This is another histological pictures of tooth germ in cup stage. This represents the oral epithelium. This is the basal cell layer of the oral epithelium, and this is the dental lamina from the oral epithelium, and this is the enamel organ during cup stage. This is the outer enamel epithelium here, and this is the cells of the inner enamel epithelium, columnar, and this is the cells of stellate reticulum. This is the dental papilla, and the outer cell is the cells of the dental sac. Here we have the enamel. We have 3 transitory structures present during cup stage and still present until the end of the early bull stage. These structures called enamel knot, enamel neck, and enamel cord. The most important one is the enamel knot because this is present in the enamel organ for the formation of the or determine the number of cusp tip in the tooth, 1 cusp or 2 or 3 cusp. This is localized cellular condensation in the region of the inner enamel epithelium here. This is the inner enamel epithelium, and the center of the inner enamel epithelium is the proliferation of cells called enamel knot. At the center of the tooth germ, it disappears in the bell stage by apoptosis. Apoptosis is mean the programmed cell death. This is the enamel organ, this is the dental lamina, and this is the oral epithelium. This is the outer enamel epithelium, and this is the inner enamel epithelium and dental papilla. Second transitor or transient structure is the enamel cord. It represents a strand of cells running from the enamel knot, from the enamel toward the outer enamel epithelium. It's in the point in the outer enamel epithelium. This is called enamel navel. When this enamel cord is continuous with the outer enamel epithelium. This causes the formation of the enamel septum, which divides the enamel organ into two parts. When there's the formation of enamel septum, there is a division of the enamel organ into two parts. This enamel organ, we have two cusps here. This is the future formed the maybe molar tooth, and this is the formation of permanent successor. This is in the beginning of the bell stage here. Third structure is the enamel-like. It is created because the dental lamina is a sheet rather than a single strand, and often contain a concavity filled with connective tissue. A section through this arrangement creates the impression that tooth germ has double attachment to the oral epithelium. So in this enamel-like represent, we have two dental lamina, but in the this is the histological section represent. This is the enamel-like. When the section is come through the dental lamina, which is like a sheet, not strand, this causes the cutting of this dental lamina into 2 or 3 strands. Between these strands is the connective tissue. This is the connective tissue represent the enamel-like. This is also transient structures, not so important, only present in the histological section. That's all for this lecture. Thank you.